We'll just start with introductions. My name is Dr. Dana Hebert-Lima, and I'm a teaching associate professor of biology. I'm an academic advisor, and I direct the Honors College Excel program. And of the hats that I wear, the one that I was actually hired to do when I got here was reinvent the molecular biology lab, the 200-level molecular biology labs and lectures for the biology department. So we'll talk a little bit about that work today as it relates to the pandemic. And I'm here with my colleague, Dr. Katrina Stewart.
Hello, how are you everybody? I'm Katrina Stewart. I have been with the biology department for several years, and in my current position, I teach the introductory biology lectures and I oversee the corresponding lab courses, Biology 116 and 118.
So just kind of by way of introduction, the biology curriculum has a 4-course series, which includes 2 100-level classes and 2 200-level classes. And the first 3 of those have a lab. So when we moved online, we had to really think about the structure of that and how many students we're seeing. So as you can see here, Biology 116 and 118 runs 2-hour labs, 24 students per lab, somewhere between 1,000 and 1,600 students total, which means we run labs all day, every day, Monday to Friday. The idea of social distancing was just completely infeasible. There's not enough time, there's not enough person power to teach any more sections of lab than we are already teaching. And the same is true for our 200-level labs, that it reaches fewer students, more like 500, but it also has a smaller lab space. We just don't have as many lab spaces. And so when the pandemic started and everything had to be moved online, we were asked pretty early what we were going to do about summer and whether online labs for an entire semester were going to be possible. And we, being the biologists that we are, thought this isn't just about summer. This is going to take us all the way through fall, and we have to design something and we have to do it quickly. So the idea of social distancing was just never going to happen for us. And so when we agreed to move the labs online, we were trying to think about how we do that while keeping the integrity of what a lab is supposed to be at the forefront. And Katrina will talk a little bit about that.
Right. So when people think about lab courses, I think they default to thinking about the practical skills, the hands-on skills that students get in labs. And certainly they do learn about those skills in lab courses, right? They get their hands on some equipment, they practice some techniques, but that isn't all they learn in labs. And there are lots of other skills that students learn in our biology labs. And when we think about the courses that we're talking about, these introductory courses, especially at the freshman and sophomore level, we really want students to be learning about what it is to be a scientist, which involves way more than just those practical skills. So we have students that are learning about scientific inquiry. They're coming up with questions, they're designing experiments, they're hypothesizing, and they're practicing all of those skills in the lab. As they are collecting data, they are then processing it. Sometimes we have them analyzing data sets that they've collected through experimentation. Sometimes they're analyzing data sets that have been provided to them. As they do that, they learn how to communicate in scientific formats. So sometimes we have them writing proposals. Sometimes we have them writing lab reports that are in the style of scientific journals. Sometimes we have them giving oral presentations to communicate what they have learned. And as they're doing this, they are retrieving information from the scientific literature and revising their work. So all of this is part of what it takes to be a scientist, and all of these things are incorporated into our lab courses. So we really wanted to think about what our students were going to get out of these courses as we were redesigning.
Yeah, and so we came at this from the perspective of backward design, but also scientific teaching. I kind of assuming that I don't have to tell this audience about backward design, except to say that you start from outcomes, right? You start from where you want students to be at the end, and then think about how you will know if they've gotten there, and then think about the activities, right? Scientific teaching is similar, except that it adds in at least in my interpretation, it adds in a layer of experimentation. So in addition to backward design, there's also this kind of implicit idea that students are becoming professionalized into the sciences to be able to think about what science is and how they practice it, but that the instructors are also experimenting, that we have ideas, we try them out, and when we get to the end here of planning experiences and instruction, we assess that piece, and then we go back again and we make sure that we're meeting our desired goals. We adjust as necessary. So the things that Katrina and I are going to talk about today are the first iteration, the first experiment that we tried with the online labs. And at the end, we'll talk a little bit more about kind of what about that is worth keeping, even as we move back into the lab space, and what about it really is the reason we have to be in lab at all.
Right. So after this moment of panic faded, right, we had weeks to design these courses and be online. We went back and looked at the learning goals. And here you're looking at a list of learning goals we have for the Biology 116 labs. And certainly we want our students to have fun. We want it to be an enjoyable experience. But when I took a look at the learning objectives that we had had from previous semesters, I realized that they were going to remain relatively unchanged. So we were still having students work both independently and collaboratively, just like they did in in-person classes. They were going to be formulating hypotheses. They were still going to be able to design experiments and analyze and interpret data. They were still going to be scientists. They were still going to be able to communicate their findings, which was important given that these are all certified. And so I was, after, again, that moment of panic, I was pleasantly surprised to re-look at these learning goals and realize, okay, this is going to be feasible. We can do this. The things that we're going to do are going to look a lot different than they did in those in-person classes, but we are still going to be able to reach those learning goals that we have established.
Yeah, and that's important for the kind of structure of these labs, because they scaffold so that students are ready for 300 and 400 level classes, right? So we can't deviate very far in terms of outcomes. We can deviate a lot in terms of activities, but we need to make sure that at the end of this course series, our students are prepared for whatever's coming next. In the 200 level labs, we do often spend more time thinking about techniques for the purpose of doing them, because there are some things that are just fundamental to what a biologist needs to be able to do. Things like moving small amounts of liquid around accurately. It seems like it would be kind of trivial, but it's really the underpinning of what most cell and molecular biologists do. Things like keeping things sterile, aseptic technique, manipulating molecules in a way that doesn't destroy them. Those are things that we could not do in the online lab. But every other piece of this found a new kind of manifestation in the online lab. So here's the version where we think about how to read and follow protocols. That is really trying to understand the molecular basis of experimental techniques. Performing calculations, reading and interpreting and producing graphs, that all becomes analyzed data produced by these techniques. And we're not collecting the data anymore, but it turns out that there are many publicly available data sets for the kind of work that we do, even including microscopy images that are out there where anybody can access them, to DNA sequences and other things that our students would normally generate themselves. But frankly, it's much more robust if we get publicly available professional data sets that they can analyze. Their data turn out cleaner and more easily to interpret when we pull from those other data sets. So there's, in what we lose from the wet lab work, and some of this technique work that we would let them practice, we're gaining in being able to find and analyze more robust, more substantial data sets.
So in order to put this in perspective, to let you know what really happened in these courses, I have some samples for you, but I want you to understand what the course themes were for both Biology 116 and 118. For Biology 116, we had a science at home theme. So students were actually planning and carrying out experiments or some sort of scientific study at home. And we also had this making connections theme. We did have an online simulation software that was available, and we would have students go through these simulations to learn about some laboratory techniques and follow up in synchronous online sessions with those as well. So Dana, let's take a look at the 118 course themes as well. So we kept that making connections theme through Biology 118, which was offered in the second summer session last summer. It's offered in our spring semester. So they were still doing those simulations, but then we also picked up a citizen science theme where students were learning about citizen science projects. They were able to participate in a project and even propose a new project. So when we were looking at the science at home for our first semester course, we wanted to emphasize to our students that it's true that we weren't going to have access to a lab. We weren't going to have access to some of the equipment that we typically have access to, but they were still going to be able to do science. It is possible to do science in your kitchen or outside or in your garage or other spaces that you have available to you. And so we tasked our students with choosing an experiment that was suitable for both their interests and their setting, what was going to be feasible for them, and had them asking questions and developing hypotheses. And they were spent some time to carefully plan out their projects. They were required to have appropriate independent and dependent variables, proper controls, and of course it had to be doable, right? So they had to really take a look at what they had available to them and make sure they could get it done in the time frame given their resources. And then of course we had them present their results in a scientific format as well. Now, some of our students were taken aback thinking, oh my gosh, I'm going to have to do this project at home. How can I do this? And so it was important for us to use our synchronous sessions to give them some peer support. So we had students do these peer-to-peer discussions a couple of times throughout the planning process. So initially when they were proposing experiments, they spent some time within their small groups in lab to talk about their questions, talk about their hypotheses, outline their experimental elements, and then their classmates were asked to give some feedback as well. And we also had students writing up these proposals in a more formal way after we had these initial peer-to-peer discussions. And then we had the students do more peer-to-peer discussion where they were really listening as the students were sussing out all of the details. And so this was important because we have students that maybe would think, okay, well, they're presenting and I'm going to sit back and listen. And so we prompted them with all of these questions as listeners to make sure that they were helping each other out, helping evaluate these experimental designs, making sure that the variables were appropriate, making sure that the students were actually measuring what they had intended to measure. And make sure that the rationale was solid as well. So I'm going to switch over and share my screen so that I can talk about another project that we did that involves some big data sets here using some citizen science. So for our Biology 118 course, this is our spring semester course, we had a citizen science theme, and we had students participating in citizen science projects, but they often aren't able to collect data. They're able to learn about the project, they're able to learn about the science behind the project, but they can't collect the data sets themselves. Those are inaccessible. However, like Dana was saying earlier, there are publicly available data sets, and we used one from Wildcam Gorongosa. This is a citizen science project based in the Gorongosa National Park in Mozambique. And this is an awesome project. It involves some trail cams that are set up in a park, and these trail cams take photographs, and the students and other participants were able to go online and screen these photos for any wildlife that they saw. And once they were screened, the information went back to the scientists running the project. Wildlife biologists were able to identify the species that were seen in the photos. And the result was a huge data set that, that they were able to analyze. So now, as our students were getting introduced to this idea, we had them do some group work in the lab. They had some tasks, and one of their tasks was to simply learn about the park as a group. Are you able to see this park map here? So this is an interactive park map, and students were able to learn about the park by clicking on it. And we had them go through a guided activity where they were able to look at limestone gorges and some different vegetation types in the park, which was important for some data analysis that they were going to do. But they could look at some other things as well. They could look at park roads and where the entrance gate was, as well as looking at the locations of the trail cameras. So they could learn about the park in this way and get a better understanding of what was happening in the park. And then we brought them back to that data set. And we were able to, from that site, download this data set. But this is a data set that is huge, right? You're looking at this spreadsheet and it's just a mass of numbers. And if I quickly go from row 1 all the way down to the bottom, you're going to be able to see that we're down here at row 40,000 something, right? So this is a huge, huge data set that can be overwhelming. And each one of these rows corresponds to a photograph. And if we go all the way over to the end, we can actually see a link for the photograph. And so this one at the top here looks something like this. And so there's an animal in this picture. And we can go back across and we can see that it has been identified as a warthog. And not only that, we can see things like how far away this camera was to water, what that water type was. We can see lots of other information about distance to human structures. And we can also see these vegetation types and when it was taken. So we can have students do some analyses. And one of the things we did when we introduced this to them was we taught them how to make pivot tables. And pivot tables are a pretty simple way to take these massive data sets and put them into something that is a little bit more understandable. And I don't know if you have used pivot tables before or not, but many of our students have not. In fact, this is for many of our students, this is their first experience with spreadsheets at all. So we had them take a look at these pivot tables and create a new one based on this data set. And we wanted them to look at some species diversity measures. And so we had them add species counts, and we wanted them to look at the numbers of the different type of species in the different vegetation types. And so with just a few clicks, we were able to get them a data set that looked much more manageable. So in these floodplains, for example, we could see all the different species that were there and how many were in that area. And so they could do some quick comparisons between the floodplains and the limestone gorges and these various other areas. Something that we did next with them was to have them brainstorm new questions. So, we started out with a somewhat guided activity to get their feet wet and get them familiar with these large data sets, but then we had them brainstorming new questions. We saw how it was possible to look at the different species, the different habitats. They could see seasons and time of day. And so we had them work in their groups to look at these possible different questions. And so, for example, during which season is the abundance of animals in the limestone gorge vegetation type the highest? If they wanted to do that, they could filter the data differently, and make a new pivot table. And please feel free to interrupt me at any time to either speed up, slow down. So based on a different question, we wanted to take a look at the numbers of species that we could see in the limestone gorges in the different seasons. And so we were able to then to take a much more manageable data set and then even have them make some basic graphs to illustrate it.
Was anything like this done prior to the pandemic in a like lecture setting, class setting, or in a lab setting that just wasn't forced upon you, so to speak?
Well, these are all skills that we have practiced in our in-person labs. So we do practice data analysis. We do practice making graphs. We do practice how to appropriately show data that will illustrate what you want it to illustrate and answer the questions. So we probably hadn't done pivot tables before, but this was a way to get at that skill in a different way. And the data sets that they're used to using were probably much smaller than this one. Dana, I'm going to turn it back over to you to talk a little bit about 220 and some of the things you did in that course.
Great, thank you. I'm going to try to share the best version of my screen here. Have I set this up in a way that you can see yourself? I was trying not to do that, but I might not be able to solve it in the time frame. So when we were setting up for Biology 220, the key inspiration came from the pandemic itself. Molecular biology is kind of the backbone of all of the research that has happened around SARS-CoV-2 in the last year. Everything from what does the virus look like and what kind of historical information do we have about that, to what is different about this version, to how do we develop treatments and how do we develop vaccines. That said, you know, although it seemed perhaps obvious to use SARS-CoV-2 as an inspiration, we were worried that students were going to be traumatized if they had to think about SARS-CoV-2 any more than they already were in their own lives, with their own families, with their own economic concerns. And ultimately, we were trying to think of a way to eventually get to what does this have to do with your life right now, without inundating them with additional kind of information and having to really like deeply think about SARS every day. So we took a slight turn and instead focused on the Zika virus. And the reasons are a little bit biological and a little bit sociological. So from the biological perspective, the Zika virus is incredibly similar to SARS-CoV-2. It is a plus-sense RNA virus. I know that there are many biologists on the meeting here, so I'm going to give more biology terminology than I might have to a different audience. They have similar kind of outside structures. They're vectored differently. So Zika is a mosquito-borne virus, whereas SARS-CoV-2 is respiratory. But they both have had recent, by which I mean in the last 10 years, epidemics with immediate real-world consequences. For our students, being predominantly from the United States, being predominantly from West Virginia, we expected that they had less experience with Zika, which presented two advantages. We thought they were less likely to be personally impacted by the Zika pandemic from kind of the 2015 to 2018 timeframe. And also we thought they would know less about it from their own experience, which meant that we could teach more about it, right? We didn't have to worry about them coming in with different assumptions about Zika because we kind of thought they might not know that much about it. And so we designed this as a series of questions. How do we visualize viruses and how do we quantify them? And the background of all of these questions is, how do we know? So the question the students have is, how does the virus interact with cells? And the question I pose back to them is, how would you know that? What technique would you need to be able to do? What data would you need to be able to collect to convince yourself that the virus interaction looks the way that it does? To be able to say the virus infects these kinds of cells, but not those kinds of cells. How would you know how that works? So that by the end of the semester, we could turn that back around and say, all those questions you had about SARS-CoV-2, you probably have the ability to answer now, because now you understand the techniques that sit behind the data that informs the news article that you read about that virus. One thing that I was a little concerned about was the fact that we run 3-hour labs. And so this just became a 3-hour synchronous Zoom session. I am exhausted by 3-hour synchronous Zoom sessions. And so I think the students would be too. And so we really chunked this out. The thing about biotechniques in the molecular space especially is that they are all derived from naturally occurring biological systems. And so there was always a place to start from lecture content. They're all in a simultaneous co-requisite lecture class that would talk about, say, DNA structure and replication, which is the basis of DNA sequencing. So we would start by saying, here's what you already know. You covered this last week in class, directly connecting it back to the lecture content. And then saying, okay, how do we repeat that skill that you learned in class? And then we would build on that and say, okay, here's a slightly more advanced version of that same idea. Here's how we've adapted it to be a biotechnique as opposed to the naturally occurring system. And then we would give them lots of data to play with. So I'm going to give you one sample activity similar to what Katrina was doing, where this happened kind of late in the semester. So we had already talked about how we look at nucleic acids. We'd already talked about how we look for proteins, and now we were talking about DNA sequencing as a set of techniques and what you could do with a DNA sequence. So we set up this activity. A patient had returned from northern Brazil with symptoms that could be ascribed to several different pathogens, including measles or malaria or dengue or probably yellow fever. When they looked at proteins, they found a positive finding for dengue. And when they looked at nucleic acids, they found a negative finding for dengue. So I would say 80% of my students are pre-med or pre-health in some way. When they look at that result, they say, well, how can that be true, right? How can it be true that it's positive for one test and negative for another? And so we say, well, what other test could we do? And that leads us into this question of DNA sequencing and what we can use for it. So we started out by giving them fake data, but raw data. That looks something like this. For those of you in the field, this is like an old school DNA gel that you would use for chain termination sequencing. And we train our students to be able to read a gel like this and determine the DNA sequence from it. Oh, sorry, I see there's a chat question. Make sure I'm not. Oh yeah, Marianne's answering the question. Thanks. The students don't get quite that far. So we train them how to read these kinds of data, also more advanced versions of DNA sequencing data like chromatographs and things like that. And then we kind of give them a walkthrough of how you would analyze that data. Now that you have a sequence, what would you do with it? One thing you can do with DNA sequence is directly compare it to anything that has ever been sequenced and put in publicly available database. So for those of you in the field, this is BLAST. And so they took the sequence that they generated from that gel and they drop it into BLAST and they see what comes up. And it turns out there are hundreds of sequences of the Zika virus that match. So then we take those sequences from NCBI, from the publicly available database, and we have them do a sequence alignment to see what's different about the sequence that they identified from their data and all of these other sequences that are already here. And I don't have student-generated data for that, but it would look something like this. So you line up all of these DNA sequences, and there are programs on the internet that you can use that say, aha, there's a difference here. where some sequences have a G and other sequences have an A. And the students say, huh, wonder what that's about. They don't actually say that, but I wish that they would. They say, oh, there's a difference here. I'll write that in my notebook. And then we take that difference and say, what does it mean though? And we put them into a different publicly available data set that looks at genome evolution. So this is next strain. If you are interested in the coronavirus, there is a next strain for the coronavirus. You could poke at a lot of data if this is your jam. But we have them look at Zika. And so they look at all of the Zika sequences that are available in this data set as a genetic phylogeny. They look at the geographical map of how the virus has spread. And you can play this from the beginning of the sequences to the end and see how they've moved. And so in this picture, it's a little hard to see. It starts way over here in Thailand. The earliest sequences were sequenced in Thailand. And then as you move along, you see that the virus mutates as it moves geographically through French Polynesia, which is over here on this map, and then eventually mutates in the population that takes over in Brazil and into North America. This giant orange bubble here is the Puerto Rican pandemic that we are probably most familiar with. And so we ask students then to go back to the data that they analyzed, because you can look at the genetic sequences down here, and their mutation was this one. So we can highlight that in this data set and see where those mutations geographically and kind of evolutionarily were sitting. So it turns out that the mutation that they identified in their data was prominent in kind of the Caribbean sequences that were identified, and then we send them to the literature and say, okay, all of this is starting to line up as a thing that might matter for physiology and for my future as a medical doctor. And when we look at the literature, we find that this single mutation contributes to fetal microcephaly, which is not the only reason people care about Zika, but is the predominant reason why people care about Zika. Most adults could get Zika and never know it, but if you were a pregnant adult who got Zika, there's the potential for fetal abnormalities. So ultimately then, we set the students up to be able to answer a lot of kinds of questions with Zika virus data, because it's publicly available and it's very well studied. And then by mid-June in the summer, we could come back to them and say, okay, but what do you want to know about SARS-CoV-2? Because that's where their minds were anyway. And so we said, all right, so you want to know, in this case, this is a sample from our students, do the mutations or the variants as they're being called in the media of SARS-CoV-2 affect severity? And they had the ability to say, how would I know that? What data could I look for? What literature could I read to be able to figure that out? And we asked them to develop something that can communicate to a 100 level student. And we picked that audience on purpose because we wanted a decently informed biological audience. So not the general public, but also not a professional audience who probably knows more about this than them, right? And so they could really be an educator in that space and think about the details they needed. So kind of, I'll invite Katrina to talk about this with me, but as we've been doing this, you know, some of the things, I think the Marianne's question from before, some of the things that I'm keeping for sure are the ability to explore techniques that we otherwise wouldn't have because they're too expensive or not robust enough to work on this number of students. We made a lab manual. It's freely available now for our students in Google. I'm pretty sure my friends at Potomac State are using some of those resources as well. So it was free to us and it was free to them. And then we have this ability now to use shared documents in a way that we couldn't before. On the other hand, you know, we have this problem with student engagement over long time periods. Our students are not going to know how to pipette when they go into upper labs, and our faculty colleagues are worried about that. And we just haven't done this enough times to be able to really evaluate how it's going. Although students were skeptical at the start, and I think that by the end they largely were on board with like, this is us making good out of a bad situation. And I think they were pretty accommodating to that. Katrina, did you have other things you wanted to say?
I don't think so. I think you summed it up well, but I'd be happy to answer any questions anybody has.
Yeah, same. I know one thing that Katrina and I have been talking about. Oh, sorry, Paul, go ahead.
I just wanted to ask, I think this is for Katrina, but in terms of the come up with your experiment at home and that sort of peer advising thing, how much that seems overwhelming in the sense of trying to fine tune people's decisions. And so I wondered what kind of variety and what some examples were of the experiments that they chose.
Sure. Well, so there were some things that I didn't include here in the slides, and they were things that we actually used with our students in lab to help them figure this out, because I wasn't sure if they were going to go the direction of, oh my gosh, this feels like 8th grade science fair, which isn't at the level where we want them to be, or completely overwhelmed. And we had more that were going, oh my gosh, I need some help getting started than I thought we were going to, but we were ready for that. And so we gave them some suggestions. So we had lots of students doing experiments with seed germination or yeast fermentation, where they were baking bread, or students that had access to kitchens could make yogurt. And all of these things can be tied back to biology and tied back to some of the themes they were learning about in lecture, including cellular respiration and fermentation and photosynthesis and all those biochemical processes. So, so those were some of the experiments that students chose to do. We had other students who were more interested in observing their environment, which was okay. So we had some students doing some basic tests on soils or water. We did have to steer students away from doing things that were simply not safe. So there were lots that wanted to do things like test out hand sanitizers and swab places that maybe have lots of bacteria and try to culture them at home. And it simply wasn't safe. And so we did do a lot of screening to make sure that they weren't doing those kinds of things. But yeah, it did take a lot of coaching. It took a lot of coaching from the TAs as well as other students through peer-to-peer. And so before they actually got started on the experiments, we had discussion posts through eCampus, where they would just put down their preliminary ideas so that both the TAs and the other students could kind of screen those to say, nah, you need to go another route, or okay, sounds good. And then as they developed their ideas, we helped them refine it. So did things like made sure their measurements and their dependent variables were actually going to capture what they wanted to know and learn, given the question that they had posed.
Did you have to do something to prevent students from converging on two similar ideas?
We tried somewhat, but we knew that there was going to be a lot of similarity given the sheer numbers. You know, we had 1,500 students, so you're going to have a lot that are interested in, putting vinegar on seeds and that sort of thing, right? So there was certainly overlap, but given all the other things that we were asking them to do after the experiment set up, we were able to kind of monitor for independent work too. They were independently writing up their findings.
Thanks. I may ask you about this offline sometime.
Yeah, sure, no problem.
Before we see if there are any other questions, excuse me, the wellness session, which is yoga with Lou Slimak, actually started at 1:20, and the next set of breakout rooms will be at 1:30. If you're not interested in the wellness center and you want to stay around, if Katrina and Dana are willing, we can continue with the discussion and Q&A aspect.
Yeah, I'm happy to stay. I was going to add to Katrina's point, to Paul's question, that you had the advantage of freshmen who didn't know each other, right? And so the space where they could cross-pollinate ideas was all built into the eCampus and other frameworks. They couldn't rely on the friends they made last year in chemistry to kind of do that kind of brainstorming, which is, I think, an advantage and a disadvantage, right? They didn't have a peer group to work with except the one that was built in.
Sorry, it looks like Marianne has her hand raised.
Yeah, go ahead.
So given the shortcoming that was the tactile portions, are you building more time into the upper level courses? Because I'll be getting some of these students later on in biomedical laboratory diagnostics, and I know we're trying to plan for that now, for giving some more time for the students for learning those skills.
Yeah, I don't know that Katrina and I can speak to that because we don't teach the upper level labs, but we are strongly encouraging our faculty colleagues to consider that, right? That assumptions you might have had about their skill set are probably faulty now, and that they will have more experience, well, at least in my case, they'll have more experience with the literature than they would have had before. They'll be better at that skill. And so time you might have spent on that probably now needs to be spent redoing aseptic technique and other things that they would have seen in my class, but they didn't this year.
Dana may have some more information on this after the fall semester. Once she's going to have the students in 220 that have just been through biology 116 and 118 both online.
Yeah, and to that point, we are thinking about that. There's two things that Katrina and I have been discussing. One is what the likelihood is that we'll be on, in person all semester, and how we might integrate our old wet labs with some small pieces of the online so that we build in flexibility, right? Just in case there's the possibility that we all have to go online, the lectures can shift pretty easily and the labs just can't with the numbers that we're running and the numbers of sections we have. And so I think to your question, we're going to build in more time for fundamentals and then leave some flexible time. If that time has to be online, we'll have that built in. And if it doesn't have to be online, then we'll have more time for fundamental skills.
Thanks for your insights in that.